Hello students, this is the help video for Lab 2.08 Investigating Biological Compounds. This lab is worth 10 points, which is equivalent to around 3 quizzes, so it's very important that you complete this lab. If you have any additional questions after watching this video and trying it on your own, you can always K-mail me or call me and I'll be more than happy to walk you through the lab. So let me give you some background information to make sure that you understand the concepts behind the lab. So in this lab, we'll be working with chemical indicators. And what chemical indicators are is they help you detect what molecules are present in different materials. So we're going to be using these chemical indicators to test what types of macromolecules are present in certain solutions. For chemical indicators, how they work is that they change color when a certain molecule is present. So they might be, for example, blue originally, but if it comes in contact with a certain molecule, then it turns into like a blackish color, for example. And so that change in color is an indication to you that there is a certain macromolecule present. So we'll be looking at different chemical indicators that test the presence of macromolecules. And the macromolecules we'll be looking at are these three right here, which are carbohydrates, proteins, and lipids. So again, how chemical indicators work is that they change when certain macromolecules are present. So right here you see the chemical indicator iodine. And iodine is a chemical indicator that tests the presence of starch, which is a type of carbohydrate. Okay, so normally iodine has this brownish color. You can't really tell from this bottle, but if you were to take it out, the liquid is actually brown. However, if you put iodine on something that contains starch, then the iodine changes from brown to a very dark black color. And if it turns into that black color, then we know that starch indeed is present. If it stays as that brown color, then we know starch is not present. So you can see this brownish color right here, even in mixed in with the milk, with the tofu and the apple. They're all that brownish color that iodine can be normally found in. However, if we mix it with starch, we see that the iodine turns a very dark color. And so that again is a positive test for starch. We have other chemical indicators for other macromolecules. We'll be looking at protein as well. Protein actually, the chemical indicator for protein are actually two things. One is sodium hydroxide and the second one is copper sulfate. You need both of them in order to test for protein. So if you were to put sodium hydroxide and copper sulfate together, it would give you this blue color. However, if protein is present, right, so if you were to mix the sodium hydroxide and copper sulfate together and put it in the presence of protein, then it would turn to this violet color. Okay, and this violet color will tell you, yes, there is protein in the solution. The last type of chemical indicator we'll be using in this lab is for lipids, and the Sudan3 is our chemical indicator for lipids. When lipids are present, it's going to turn this bright reddish orange, so you're going to see like a bright color, whereas if lipids are not present, you'll see more of this dark color, which you see right here. So all three of these chemical indicators undergo some sort of change when they are in the presence of a certain macromolecule. If there's no change in the chemical indicator, again, that shows you a negative test for that macromolecule. So here's a summary of the three chemical indicators. We have one, iodine, which tests for starch. Usually it's like a dark red or brown color, but if it's in the presence of starch, it turns black. The The next indicator we talked about was copper sulfate and sodium hydroxide together. It tests for proteins. If there are no proteins, it'll stay its bluish color, but if it's a positive test, then it'll turn violet or a dark purple, almost looking black. And the last one is Sudan-3, and that tests for lipids. Normally it's a dark red color, so if there's no lipids present, then it'll remain dark red, but if there are lipids present, then it'll turn a reddish orange, which again, remember, is very bright. Okay, so in the lab, you'll be testing the following items to find out what macromolecules are present. We'll be testing water, milk, and an unknown solution. Obviously, water does not have any macromolecules. So when you test water with Sudan-3 and the other two chemical indicators, they should all be negative tests. So you might be asking, why exactly do we even test water then when we know that all of them are going to be negative? They act as our control. Okay, so water is our control to make sure that all the chemical indicators are working properly. If for some reason we put the chemical indicator in water and there's a positive test, then that means that the chemical indicator is not a reliable chemical indicator to tell us whether a certain molecule is present or not. Okay, so water will be our control. Okay, so the lab report for Lab 2.08 looks like this. I put the summary of the chemical indicators at the top so you can use it as a reference. You'll need to fill this out as you go through the virtual lab, which I'll show you in just a second. So what you see here is, first you're going to test the water, the milk, and the unknown solution. You're going to test it using iodine, copper sulfate, as well as Sudan-3. In these boxes, you're going to go ahead and tell me what color each test ends up being, and whether there was a change or no change. Okay, so that would be the best way to answer each box. Tell me what color you see and whether there was a change or there was no change in the chemical indicator. Okay, so you're going to do that for all of these boxes right here. Based on what the data that you get from the first question, you're going to answer and form some conclusions about what was in the unknown solution and in the milk. Okay, so in the unknown solution, you're going to answer what was present. You could say, in the unknown solution, there was whatever macromolecule present. Okay, in the unknown solution, you should have one macromolecule present. So if you got negative tests for all of them, then you need to redo it. Okay, and then how do you know what was present in the unknown solution? You can say that the chemical indicator changed, right? And because the chemical indicator changed, you know that that macromolecule is present. And the same thing for milk. The milk, you want to put in the milk solution, there was present. And there actually are two macromolecules that are present in milk. So make sure that you're able to see two of them. Okay, and you'll describe how you know that as well. The next question is asking, what was the role of the water test tube in each phase? Remember, we just talked about that. We know that water will always yield a negative test. However, we use the water as a control. Okay, remember, explain this in a complete sentence. Right? And the last question is going to ask you, describe a chemical indicator. So you want to describe what a chemical indicator is. So what is it? What is it used for? And how does it work? Okay, so these questions you should be able to answer based on the slides that we just went over on the PowerPoint. All right, and then I'll give you a total of 10 on this lab. All right, so it's important for us to be able to fill out this data right here. So we're going to go ahead and go over the lab right now. All right, so if you log into the course page, in order to get to the lab, you can click on 2 under class content, scroll down until you reach 2.08, and then you want to click on investigation, investigating biological compounds. Okay, once that loads, you want to go ahead and click on the right arrow so that you go to page 2 of 2, and you can click on investigating biological compounds. It's going to tell you to select the link below, so you go ahead and select it, and then you can press begin the lab and then choose a glove. And press start. So remember, we're going to test the unknown solution, the milk solution, as well as the water solution for our data table. Okay, so in order to figure out what happens when you put in water, you first need to take the water, and put it into all of the water tubes. So you see starch control, which is water, protein control, which is water, and don't forget the lipid control as well, which is water. And then you can go ahead and put that back. To make sure that everything is working properly, we want to make sure to test the chemical indicator, and something we know would also give a positive test. So remember, for starch, right, if we put starch in here, iodine should change color. So we could take a drop of starch and put it in there. And then we can go ahead and take iodine and put it in the water and in the starch. So if we were to put it in the water, we expect no change because there's no starch in the water. So you see it still has that brown color. Okay, so in your data table here, you could put that it had, it has a brown color, and that the color did not change. Right? But if we were to put iodine into starch, we see that it turns into that dark purple or black color. Right? Now let's go ahead and test for the protein. So the protein sample we'll use is albumin. So you can take a drop of that and drop it in. And then remember, our chemical indicator for protein consists of 2 things, sodium hydroxide and copper sulfate. So I'm going to first put sodium hydroxide into both of them. Right? And don't forget copper sulfate. So remember, typically, if it doesn't have any protein, then it'll just be blue, which you see right here. Water doesn't have any protein, so it's just blue. But if it does have protein, then it turns that dark violet or black color, which you see right there. And because we know albumin is a protein, we see that it's a positive test. Okay, but we look at the water one, and there's no change. So we could say it's light blue, and there was no change in the color. All right, the last one is for lipids. To do the lipid test, we're going to use vegetable oil and put it in this tube. Then we will use Sudan-3, which is our test for lipid, our chemical indicator. We see that it still has that dark reddish orange color. But if we put it into the vegetable oil, we see that it turns to a more bright color. Hopefully you can see that difference. This one is more like a dark, dull color, and this one is more bright. But we see the water did not change, so we can go ahead and put that into our data table as well. We'll say that it's a dark orange red, and there was no change. Okay, so that's how you fill out your data table. You need to do that for milk and the unknown solution as well. So I'll help you set up both of those, but you should complete it on your own. So for the unknown solution, the first thing you need to do is you need to put the unknown solution into each of these tubes. Once you put the unknown solution in each of these tubes, then you will put the chemical indicators. So the unknown for starch, right? Then you'll put iodine in here. The unknown for protein, you're going to put sodium hydroxide and the copper sulfate in here. And then the unknown for lipid, you'll put the Sudan-3 in here. And you'll see, is there a color change or not? And that'll tell you whether certain macromolecules are present. And you'll do the same thing for milk. For milk, you'll go ahead and take the milk dropper and drop milk into each of these. Once you do that, then again, for the milk starch test, you'll put iodine into the milk starch. For the protein, you'll put the sodium hydroxide and the copper sulfate into the milk. And the last one for lipids, you'll put Sudan-3. If there's a color change, that means that macromolecule is present. If not, then it is not present. So you'll go back here, tell me which color is present in each tube, and whether there was a change or not. If you're confused about whether there was a change or not, you can look up here, right? Is it a negative or positive test? The negative test means there's no change. The positive test means that there was a change. Okay, and then using that information, you can then answer the following questions as well. Okay, so that's an overview of the lab. If you have any additional questions, you can always K-mail me or call me and I can walk you through it. Remember to submit your lab in the Dropbox. You'll go to Tools, you'll go to Dropbox, and then you can go ahead and drop it into the 2.08 Investigating Biological Compound. And I'll go ahead and grade it as soon as I see that you've submitted it. If you need help finding the lab, you can go to, Content. You can go to 2 and then 8. Once you're at 2.08, you'll see the Digital Materials Lab 2.08 report. You can click on that and download it as well. Okay, again, if you have any additional questions, please feel free to contact me and I'll be more than happy to help walk you through it.